Welcome, my dear viewers, to Biology at the Red Priyajit. In today's class, we will explore the structure and functional significance of the nucleus with special emphasis on mRNA transport through the nuclear pore complex, the organization of chromatin, and the structure and function of the nucleolus. This session will help you understand how genetic information is packaged, regulated, and efficiently transported from the nucleus to the cytoplasm. Transport of mRNA through the nuclear pore complex. Introduction. In eukaryotic cells, transcription occurs in the nucleus where translation occurs in the cytoplasm. Therefore, newly synthesized mRNA must be transported out of the nucleus, and it occurs through a specialized structure called the nuclear pore complex, or NPC. This is the image showing the structure of nuclear pore complex. In presence of nuclear pore complex, the transport process is highly regulated, energy dependent, and it ensures that only properly processed mRNA can reach the cytoplasm. So, nuclear pore complex overview. NPC is basically large multi-protein complex about 125 megadalton in vertebrates. It is composed of 30 different proteins called nucleoporins or NUPs. It forms a selective channel for transport between nucleus and cytoplasm. So the transport rules: if the molecules are smaller in size and if it less than 40 kilodalton, it pass by simple diffusion. But if the molecules are larger, like mRNA protein complexes or mRNPs, it require active transport. Formation of export competent mRNP. Before export, mRNA must undergo proper processing, and the processing steps include 5' capping, splicing, and 3' polyadenylation, binding with RNA binding proteins. These steps together form an mRNA protein complex or mRNP. Transcription, splicing, and processing to form a mature mRNP. And when formation of an export competent mRNP, then docking and translocation occurred through nuclear pore, and the nuclear transport causes the cytoplasmic remodeling of the mRNPs. Protein assembled on mRNA, cap binding complex or CBC, it binds with the 5' cap. Exon junction complex or EJC, it added after splicing. HNRNP proteins, they help in packaging and stabilization of the mRNA. TREX complex, it links transcription with the export process, and export receptors include NXF1 or TAP and NXT1 or P15. Importance: only fully processed mRNA is export, and it prevents translation of faulty RNA molecules. Role of TREX protein. What is TREX? TREX is basically transcription export complex recruited during the transcription elongation process. What is the function of this complex? It couples with the mRNA synthesis, processing, and export. It also recruits NXF1 and NXT1 export receptor protein. Major components include UAP56, ALI, REF complex, Rho complex, DDX protein, etc. And in absence of the TREX complex, mRNA accumulate in the nucleus, and they cannot move or cannot be transported from the nucleus to the cytoplasm for the further translation process. So export receptors include the NXF1 and NXT1. They are the main export receptor for most mRNAs. Exist as a heterodimer. So NXF1 or TAP binds mRNA, and NXT1 or P15 stabilizes the complex. It interacts with the FG repeat, the nucleoporins of nuclear pore complex, and it replaces many heterogeneous nuclear RNPs and escorts mRNA to move through NPC. Docking at the nuclear pore complex, export ready mRNP is guided to nuclear pore complex by the adapted proteins. Then mRNP docks onto cytoplasmic fibrils of NPC. Inside the pore, FG nucleoporins form a selective permeability barrier, and mRNP passes through NPC in a directional manner. Translocation through the NPC. During the passage through this pore, nuclear pore, NXF1 or NXT1 repeatedly binds FG repeats, allowing stepwise movement. What is FG repeat? F is an amino acid and G amino acid. This is your task to identify which there is a one letter code of the amino acids, so you must identify F indicates which amino acid and G indicates which amino acid. So FG repeats is meaning is the repeats of those two amino acids. And mRNA remodeling occurs, some proteins detach and others are attached. ATP dependent helicase DBP5 assists the process and they present in the cytoplasmic site. Cytoplasmic release and remodeling. Once RNP reaches the cytoplasm, DBP helicase activated by GLE1 and inositol hexa kiss phosphate IP6 remove NXF1 and NXT1 from mRNA. Final remodeling of mRNP occurs. Then CBC is replaced by EIF4E and preparing mRNA for translation. It exports factors which are recycled back to the nucleus, and this irreversible removal ensures unidirectional export process. Energy requirement: the export does not require GTP hydrolysis unlike protein is import. Energy is required for DBP5 ATPase activity, mRNP remodeling, transcription and RNA processing. So overall the process is ATP dependent and it is an energy requiring process. Quality control mechanism of this process, we know that only correct mRNA will be allowed to exit from the nucleus, and there are checkpoints. Checkpoints include the completion of the splicing event and proper 5' capping and also the 3' poly A tail. Correct assembly of TREX protein complex and export receptors must be done. The defective mRNA handling, from the image we can show that retained the mRNA should be retained in nucleus by retention factors and it should be degraded by nuclear exosome complex, protein complex, and it prevents the synthesis of incorrect proteins. So, what is the summary of the mRNA transport? It occurs through nuclear pore complex. It requires export competent mRNP or messenger RNA protein complex. TREX protein links transcription and export process. NXF1 and NXT1 directs the mRNA to move through NPC. And FG nucleoporins protein provides selective passage through the nucleus. DBP5 ensures cytoplasmic release. Only fully processed mRNA reaches the cytoplasm. This is the image showing the mRNA transport process. And now we should study about the nuclear matrix or nuclear sap or karyolimb or the nucleoplasm. This is the image showing the nucleoplasm. So it is a transparent semi-solid matrix present during interphase. It is also known as karyolimb or enchylema or nuclear sap or or nuclear matrix. It is composed mainly of proteinaceous material. It acts as the site of enzymatic activities, shows variation during cell division. It may contribute to spindle formation in plants. RNP structures in nucleoplasm. Barnhart, 1968. Interchromatic RNP fibrils, interchromatic RNP granules, perichromatic RNP granules. They are approximately interchromatic RNP granules are 250 angstrom and perichromatic RNP granules nearly 450 angstrom. And the RNP coiled bodies and RNA granules are 300 to 400 angstrom. Nucleolus, it is dense, rounded, non-membranous body found inside the nucleus. They first described by Fontana in 1781. They are acidophilic and the clearly visible complex. They disappear during prophase and reappears in daughter nuclei. Components, parts amorpha, the amorphous and it disappears first during nuclear division. Nucleolonema, this is a permanent filamentous structure and it contains RNP particles. Types of nucleoli: E.B. Wilson. Plasmosomes, they are oxyphilic, they are transparent to halo with dense core. Karyosomes, they are chromatin nucleoli, they are basophilic, they are associated with chromosomes. Types, they are net knots like strike, chromosome nucleoli are there, or karyosphere. This is the image showing the nucleoli structure. MP nucleoli, it is combination of plasmosome and karyosome. They common in eggs of mollusk, annelids, etc. Ultrastructure of the nucleolus, it has granular component. These are the image showing the granular region. Ribosome precursor particles are there. Fibrillar region, this is the fibrilla center. 50 to 80 angstrom region. rRNA genes and nascent rRNA genes are there. Amorphous region, this is the low electron dense region. Nucleolus associated chromatin, it is found surrounding the nucleolus. So what is the functions of nucleolus? They role in mitosis, they help in ribosome synthesis, in protein synthesis, and they involve in transmission of genetic information. So the chromatin DNA complex, this is the DNA, red colored DNA, and there is the histone protein with a component of chromatin. So DNA complex with histone and non-histone proteins in the chromatin, and histones are basic proteins rich in lysine and arginine amino acid. Core histones include 4 types of proteins such as H2A, H2B, H3 and H4. H1, this is the green colored is the H1 protein that acts as linker DNA. It binds linker DNA and stabilizes the higher order structure of the nucleosome model. This is nucleosome. So types of chromatin, this is the higher order structure of the chromosome. Types of chromatin, heterochromatin which is darkly stained and genetically inactive. Euchromatin is lightly stained and transcriptionally active. So packaging of chromatin, it is proposed by Roger Kornberg in 1974. DNA wraps around the histone octamer, the image showing the wrapping of DNA surrounding the histone octamer. It is known as nucleosome model, and then it forms a beads on a string. This is a beads on a string structure, or known as 10 nanometer fiber. Further it will form 30 nanometer fiber or solenoid or zigzag model. This is solenoid or zigzag model or 30 nanometer fiber, and ultimately nucleosome plus H1, it called the chromatosome. So what is the functions of nucleus? It controls cellular metabolic activities, store genetic information. It is responsible for the formation of ribosome. It controls cell division. It induces genetic variation. It brings about cellular differentiation. So thank you, my dear students, for joining today's class. To conclude, the nucleus is a highly organized and dynamic organelle essential for cellular regulation. The nuclear pore complex ensure selective mRNA transport. Chromatin packaging allows efficient DNA organization and gene regulation, and the nucleolus plays a crucial role in ribosome formation. Together, these nuclear components coordinate gene expression, making this topic fundamental for undergraduate and postgraduate level cell biology studies. Stay curious, keep learning, and see you in the next class. Thank you.